As we saw in the previous section, Bernard Katz and his contemporaries used electrophysiology to provide convincing evidence that neurotransmitters were released not as a steady stream, but instead in discrete packets. They hypothesized the existence of structures called vesicles that were loaded with a set amount of neurotransmitter molecules. However, they had no way to visualize these vesicles, so there was no conclusive proof of the vesicle hypothesis. After all, seeing is believing. For many years, researchers searched for the technology needed to peer into the synapse. Light microscopes don't have the resolution needed to study the structures inside of the synapse, many of which are only a few nanometers in diameter. Electron microscopes provided the solution to this challenge. Modern electron microscopes can resolve objects that are hundreds of picometers across, while the best that a light microscope can do is several hundred nanometers. Viewing the synapse with electron microscopy revealed the presence of hundreds of small circular structures at the presynaptic terminal. Additionally, researchers observed that by imaging right after excitation, they found omega bodies, named for the shape of the vesicles that formed when they fused with the membrane of the presynaptic terminal and released chemical payload into the synaptic cleft. Lastly, it was observed that after heavy stimulation of a neuron, the presynaptic terminal was actually depleted of these structures. This suggests that they were consumed and needed to be regenerated. Together, these images confirm that there exists a physical structure, the vesicle, that could explain the results observed by electrical recordings. But, as with many technological breakthroughs, electron microscopy not only confirmed older hypotheses, but allowed a whole new understanding of the synapse. For one, it provided important spatial information that you simply cannot find with electrical recordings. Researchers observed that there were two distinct populations of vesicles, ones that were docked and ready to go right behind the presynaptic membrane, and another pool that was farther back, waiting to be loaded and docked. The insights gleaned from using the electron microscope to image the brain are not limited to the visualization of vesicles. Today, we have even more powerful scanning electron microscopes that are being used to visualize circuitry and map the complexities of cell types and interactions in the brain tissue at a very high level of resolution.